Okay, Assalamualaikum. Berjumpa lagi kita untuk second video. So, for this subtopic, we will discuss about the general characteristic of enzyme and the extracellular and intracellular enzyme. Okay, so stay tuned. Okay, now let we discuss about the general characteristic of the enzyme. So, in regarding or referring to our textbook, says about 8 general characteristics of enzyme that you must know. So, the first one is the enzyme act rapidly, mean instantly. When we use the enzyme, so it will start working on that time. So, the second one is the enzyme are biological catalyst that speed up. The key point is speed up the biochemical reactions. For example, I give you one example which is the detergent usage in our daily life. Let's say kalau kita basuh baju dengan detergents and another one is without detergent. So, the stain, kotoran itu akan lebih mudah hilang dengan menggunakan detergent. So, kamu kena faham yang detergent itu dia mengandungi enzyme. So, that's why for certain types of detergents ini digunakan, dia ada dia punya specialist dia, iaitu dia boleh menanggalkan kesan kotoran dengan lebih baik semua kan. So enzyme itu dia mesti ada lipase. Sebab apa dia nak remove the stain, the types of lipid tadi, kesan kotoran makanan, minyak dan sebagainya. Okay, the third one adalah some enzyme need cofactor to work more efficient. Cofactor just like pembantu. Means dengan kehadiran cofactor ini, contohnya vitamin B dengan magnesium iron, for certain enzyme ini, dia punya tindak balasnya akan menjadi lebih cepat dan lebih efficient, dengan lebih bagus. Next adalah, the enzyme are only required in small quantities. Diperlukan dalam kuantiti yang sedikit and are reusable. Okay. This is the example if you refer to the commercial TV punya iklan kan, you can see which is dia ada kalau iklan detergents tu, Dynamo ini lebih berkesan menanggalkan kotoran berbanding dengan serbuk pencuci X, right? So walaupun satu penutup itu, dia punya kuasa dia untuk bersihkan kotoran itu adalah bersamaan dengan 4 sudu ataupun 4 penutup sabun jenama X. So this is mean the enzyme inside the dynamo is a very powerful, which is just needed in a small quantities, and then it is reusable. Reusable means if you have complete your wash, the first one you can use the same water that contain the detergents for the second wash. That's why if you see in the kedai makan kan, untuk kedai makan di mana bila dia basuh di pinggan itu, dia akan ada satu besen tu yang mengandungi air, yang air kemudian dia ada sabun, and then bila dia bilas itu, and then baru dia akan masukkan dalam satu besen lain pula kan. Itu menunjukkan enzymes are reusable. And then right, so the structure of the enzyme is remain unchanged. Means after the reaction, the structure of the enzyme remain unchanged. Dia tidak akan berubah bentuk and are not destroyed after the reaction, which is it is remain the shape of the enzyme and it can work for the next reaction. Next, if the reactions of enzyme are specific, mean the lipid only will work on the lipase only work on the lipids only. Means lipid tidak akan ditindak balas oleh enzim yang lain. Contohnya amylase or protease or whatever kan. So dia tidak akan bertindak balas ke atas lipid. So hanya lipase yang akan bertindak balas ke atas lipid. Right. Kemudian, we can see that most of the reaction catalyzed by enzyme are reversible. Bermaksud dia boleh reversible lah. Dia boleh catabolism dengan anabolism tu. And then next one adalah enzymes activity can be slowed down or stopped by the enzyme inhibitors. For example, is a heavy metal such as lead, plumbum dan juga mercury. So inhibitors ni maksud dia dalam bahasa Melayu adalah perencat, which is it have the same active site, the shape of the active site of the inhibitors same with the specific ataupun enzyme tersebut. Di mana bila katakan tapak perlekatan ataupun active site of the substrate itu sama match dengan inhibitors nanti, so inhibitors dia akan bind dekat kawasan dengan substrate tersebut. So what will happen is the enzyme will not break down the substrate into the product. So for certain reaction tidak akan berlaku. That's why mercury is very dangerous during our class, during lab. So the teachers will keep always remind you, be careful with the thermometer because the mercury alright, so for the next one is about the types of enzyme. So the types of enzyme, we've got two types. The first one is intracellular enzyme and the second one is extracellular enzyme. Okay, untuk mudahkan kamu to ensure to make it easier, intracellular means synthesized in the cell and used for the cell itself. Let's say lah contoh, kalau kita masak nasi, kita gunakan, dia akan dimakan oleh orang di dalam rumah kita. For extracellular enzyme, which is synthesized in the cell but secreted out from the cell to work externally. Maksudnya, in other words, yang cikgu nak mudahkan kamu adalah kamu ingat, okay, yang ni contohnya kamu masak nasi tapi bukan untuk diberikan untuk dalam orang rumah kamu, tapi akan dihantar keluar sebab kamu jual nasi tersebut. Itu maksud dia. Okay, so from here you can see that intracellular enzyme, the first one you ingat, is written in the cell for the use of the cell itself. Basically found in the cytoplasm, nucleus, mitochondria and the chloroplast. For example, as you can read here, which is contains the hexokinase enzyme in the cellular respiration. The process that involved here is the glycolysis. You see, the glyco means glucose and lysis means the breakdown. So the breakdowns of glucose will involve the hexokinase enzyme to produce energy and pyruvate. The second one is extracellular enzyme. This one adalah the productions of the trypsin enzyme. Let's say by the pancreatic cell, but then it will secreted into the duodenum to break down. Okay, we will discuss about the productions of extracellular enzyme. Well, it will involve several cellular components, which is which are from a nucleus, mitochondria, rough endoplasmic reticulum, ribosomes, transport vesicles, Golgi apparatus, secretory vesicle. So there are two types of vesicles here. You must remember, the first one is transport vesicle, the second one is secretory vesicles. Transport vesicles mean it will transport the protein from the ribosome to the Golgi apparatus. But from the Golgi apparatus to outside of the cell, it will use the secretory vesicle, vesicle rembesan. Yeah. To understand this subtopic better, you must understand when we discuss this topic, we must refer to the diagram. Okay, let's say the first one, we will starting from the ribosome, which is the site of protein synthesis. The second one is, we look at the number 2, which is the protein synthesized by the ribosome at the lumens of the RER, rough endoplasmic reticulum, and will transport it through it. Next is the third one is once it reached at the end of the RER, the membrane buds off to form a transport vesicle. Mean how the protein will be transported out from the rough endoplasmic reticulum is by using the transport vesicle. So this transport vesicle contain proteins that will move towards the Golgi apparatus, then it will fuse into the Golgi apparatus. So the fifth one is the Golgi apparatus, which is the protein that are modified into enzyme and secreted in the secretory vesicles, and that are formed from the tip of the Golgi apparatus. So the functions of Golgi apparatus, you must remember, it will pack, modified, and transport the protein. And the sixth one, the last one, is the secretory vesicle will move towards the plasma membrane and fuse. Okay, this is also the flow chart, which is shows the productions of extracellular enzyme. As I mentioned before, first one, protein itu disintesis di dalam ribosome. For the second one is the protein yang telah disintesiskan tadi akan masuk ataupun difuse masuk ke dalam rough endoplasmic reticulum dan diangkut melaluinya. And then the third one, which is at the end of the rough ER, bila dia sampai dekat penghujung ER tu, dia akan budding off. Budding off means dia akan menunas, dia akan bertunas keluar to form the transport vesicles. And then lepas itu what will happen, the transport vesicles that contain the proteins, dia akan move towards, bergerak ke arah Golgi apparatus dan fuse dengannya. Next slide will show the continuous flow chart, which is the protein further modified. Dia akan diteruskan dengan dia punya ubah suai untuk jadi enzyme dalam Golgi apparatus tersebut. So the next one is the protein that will wrap in the circulatory vesicles. Dia akan dibalut, dia akan di-wrap di dalam vesicle rembesan. So this vesicle rembesan, dia akan bud off, dia akan tunas keluar daripada Golgi apparatus tadi and then travel to the plasma membrane. And then after that what will happen? The vesicle will fill the plasma membrane before secreted as the extracellular enzyme. Okay, so that's all for this video. So stay tuned for the next one. Thank you guys.